Good day, this is the Professor, CBMD PhD, and today's Tuesday, September 26, 2023. It is 3:03 and 40 seconds, AM Eastern Standard Time, and today's research, dendritic cell in cancer. So let's hop to it here. A dendritic cell vaccine was safe and induced immune responses in patients with multiple myeloma. Vaccination given in combination with autologous stem cell transplant was associated with long-term disease control. Date September 22nd, 2023. Source: American Association for Cancer Research. Autologous stem cell transplant is typically preceded by induction therapy with chemotherapy to kill as many cancer cells as possible and induce a remission. For this study, the investigators selected patients with high-risk disease who still had active myeloma after induction therapy and before receiving autologous stem cell transplant, which constitutes a negative prognostic factor. They focused on this patient population because they are most in need of the vaccine, as dendritic cells are an essential component of the immune system. They take up foreign proteins, break them down, and present the fragments, peptides, to other immune cells to stimulate the immune response. And to produce the vaccine, the researchers engineered the patient's own dendritic cells to express cervivin and induce an immune response against the protein. And high expression of cervivin at diagnosis is associated with poor outcomes. Therefore, they hypothesized that by targeting this protein, they could induce an immune response in patients who have the most aggressive disease and potentially keep them in remission for a longer period of time. The scientific investigators and microbiologists designed a dendritic cell vaccine targeting a protein called cervivin and tested this vaccine in a phase 1 clinical trial involving 13 patients with multiple myeloma. Patients received one dose of the vaccine within 30 days before standard of care autologous stem cell transplant and another dose approximately 21 days post-transplant. And to increase the number of cervivin peptides presented in the immune system and therefore the likelihood of triggering a cervivin-specific response, the team engineered the dendritic cells to express a version of the entire protein with a mutation to increase safety without compromising immunogenicity. The vaccine in combination with autologous stem cell transplant was well tolerated, with only minor adverse effects observed. Furthermore, the vaccine induced cervivin-specific immune responses, specifically circulating cervivin-specific CD4 T cells and CD8 T cells, significantly increased approximately 35% and 30% of patients, respectively. Antibodies against cervivin peptides were detected in 2 out of 13 patients at baseline and 9 out of 13 patients after vaccination. The autologous stem cell transplant, overall 85% of patients had either a T cell response or an antibody response against cervivin, and 7 patients experienced an improved clinical response at 9 days post-transplant, all of whom showed cervivin-specific immune responses after a median follow-up of 4.2 years. 6 out of these 7 patients were alive and remained disease-free after treatment. The estimated 4-year progression-free survival was 71%. These results compared very favorably to historical data, suggesting the 4-year progression-free survival of this patient population to be approximately 50%. Larger randomized studies are needed to confirm the findings and to assess whether moving vaccination to earlier in the disease course could be beneficial in preventing patients from developing aggressive forms of myeloma. The patient population in this study is not often included prospectively in clinical trials. Furthermore, the treatment landscape for myeloma is rapidly evolving. The new generation therapies, such as CAR T-cell therapy and bispecific antibodies, being tested before transplant. Because of these methods, there are limitations to directly comparing the results from this study to historical data. In addition, limited sample availability did not allow the researchers to confirm that the observed vaccine-specific immune responses were directed against the patient's own myeloma cells. And this is the professor, professor of microbiology, bioinformatics, pathophysiology, and later of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.